Hello, I'm Dr. Nicola Annels, Senior Research Fellow from the Targeted Cancer Therapy Research Group at the University of Surrey. My research interest is understanding the immune biology of cancers so that we can develop effective immunotherapies for cancer patients. I'm going to outline for you today the importance of using state-of-the-art technologies for cancer immunotherapy research and give you some examples of the types of technologies we are using and that are benefiting the research in our lab. Firstly, it is important to understand that cancer cells do not exist in isolation, but rather that a tumour is made up of not only cancer cells, but multiple cell types, including many different immune cell types. The dynamic interactions of cancer cells with their surrounding tissue and different immune cell types can result in the cancer cells controlling their environment, resulting in uncontrolled growth by the cancer cells and evasion of the immune system. To fully understand this phenomena, we must go beyond the traditional methods of culturing cancer cells in 2D monolayers or staining cancer tissues with just one or two biomarkers, and rather use technologies that allow us to really understand the complexity of the tumour microenvironment. Immunotherapy treatments rely on the activation of the patient's immune system to recognise and kill cancer cells. The key to understanding more about the cancer immune biology is in studying the complexity of the tumour microenvironment. One approach that is widely utilised at the University of Surrey is the use of multiplex immunofluorescent staining. This allows us to stain up to 8 different markers on a single 4 micrometer thick section of tumour tissue, allowing us to analyse multiple different immune cell types and markers. This technique allows us to accurately quantify the different immune cell types present within a section of tumour, as seen in the top right-hand side of this slide, which shows immunofluorescently labelled cell types within a section of bladder cancer. Furthermore, we can visualise how the immune cell types are spatially organised within the tumour tissue and how they interact with each other and with the tumour cells, depicted here in white. Using this multiplex approach, we can actually look at the immune cells and their markers and understand whether these immune cells are activated or whether there are immunosuppressive cell types nearby that might be inhibiting an anti-tumour immune response. This information will allow us to reliably predict treatment response or disease progression through the development of biomarker signatures, as well as to help us develop novel immunotherapeutic strategies. Another technique that we consistently use alongside multiplex immunofluorescent staining is the use of nanostring 360 gene expression profiling. This unique 770 gene expression panel allows us to profile the tumour tissue of interest for vital components involved in the complex interplay between the tumour, the microenvironment, and the immune response. This provides researchers such as ourselves with a comprehensive characterization of disease biology and an understanding of the different mechanisms the tumour uses to evade the immune response. Sometimes in the lab, we receive fresh tissue direct from surgery, and using an instrument called an octodissociator, we are able to dissociate the tumour tissue into a single cell suspension. These cells can be fluorescently labelled and run through another instrument called a flow cytometer, which allows us to identify cancer cells or the different immune cell types that were originally present within the tumour tissue. Finally, as I said before, we're moving away from the traditional methods of culturing cancer cells in 2D monolayers. In the body, tumour cells do not exist as single entities, but rely on cell-to-cell communication across different immune cell types and with their surrounding matrix and tissue. This tumour microenvironment regulates the growth of cancer cells, their invasion and metastasis. Thus, to truly mimic the complex and dynamic interactions of the tumour microenvironment, we have adopted multicellular 3D models in the lab with which to test our anti-cancer agents. On the left shows our ability to grow tumour-derived organoids. These are 3D cellular clusters derived from cancer stem cells that mimic the original tumour characteristics and cellular composition. We are able to monitor the growth of these tumour organoids or to look at the effects of anti-cancer treatments on these tumour organoids in real time using an instrument called an IncuSite. The IncuSite is a live cell imaging technology that is specifically designed to fit within any incubator. This technology is ideal for long-term experiments where we're treating cancer cells with cancer-killing viruses or anti-cancer agents that require long-term monitoring. A second example of the use of 3D models that we utilize is precision cut slicing of tumour tissue. Upon receipt in the lab of this fresh tumour tissue, direct from surgery, we're able to embed the tissue in agar and cut this using an instrument called a vibrotome. This allows us to culture viable tumour tissue in the lab, keeping the exact architecture of the original tumour whilst testing our anti-cancer agents. So from this brief overview, I hope you can appreciate the importance of funding these state-of-the-art technologies for cancer immunotherapy research. The knowledge gained from the use of these state-of-the-art technologies will help to identify targets to disrupt tumour cell interactions within the tumour microenvironment and help to develop safe and effective immunotherapies to fight cancer.